Welcome listeners to Talking Eyes. I'm your host Leanne Trin, an optometrist and public health clinician based in Larrakia Country, also known as Darwin in the Northern Territory of Australia. This podcast was created to translate exciting innovative developments from vision research into a form that both clinicians and really any curious mind can understand. Innovation, simplicity, clarity. This series will be diving into a topic where the research has been absolutely awe-inspiring: the retina. It's just a small part of the human eye, roughly the size of a postage stamp, but it's an incredible piece of human biology that plays a central role in how we experience the world around us. Imagine this for a moment. You're as lucky as I am to be sitting on the beach, admiring a stunning Larrakia Country sunset, and all those brilliant wavelengths of light scattered throughout the atmosphere, filling your vision with hues of orange, red, purple, pink. A flock of magpie geese cross the brilliant sky on their way home to roost. You gaze out at the beautiful ocean, perhaps sitting with your partner, watching your children play near the waves, lapping into the dripstone beach, recognising in the sands imprints where ocean reptiles have been. All of these magical moments, whether they're about appreciating colour, reading the scene, recognising your loved one's face and movements, they just would not be possible without your humble retina. What exactly is the retina? I asked some colleagues how they might describe this complex bit of tissue to their patients.
Okay, the retina is at the back of the eye. Light passes through our eye and hits the retina. The retina consists of layers of cells that help convert the light into the images that we see.
The retina at the back of the eye, there are four main areas that we pay attention to. So firstly, there is the optic nerve head, which is like the pale circle or oval, and this sends the visual signal of what we are seeing through to the brain. The second area that we look at is the macula, which is the dark area near the optic nerve, and we use this part of our retina for central vision.
In the middle of the retina is the macula. The macula is responsible for our central vision. Right now, you are looking at me with your macula. We also use our macula for reading and recognising faces. You may have heard of macular degeneration. It's a common eye disease that usually affects older adults.
We also look at the blood vessels, so the arteries and veins, and look for any changes that might be a sign of high blood pressure or diabetes. And then lastly, depending on how we're looking at the retina, we can look at the peripheral retina, so the outer area of the retina. And there are some eye diseases that would affect or damage this part of the retina before the more central areas.
The peripheral retina is thinner than the central retina. This makes it more susceptible to retinal tears, holes, and detachments. Flashing lights in your vision, like a speeding camera flash or like lightning, can be a sign of a retinal detachment. See your optometrist if you notice these. Also, any changes to your floaters are also worth mentioning to your optometrist.
We are going to unpack as much as we can about the structure and function of the retina so that when you hear from our experts in the field in our future episodes, we have a really clear understanding of the areas being targeted, how they're being targeted, and why they're being targeted. Today, I have with me my inaugural guest, longtime friend, incredibly talented brain and co-designer of this particular series, Talking Retina, Associate Professor Lauren Ayton. Hi, Lauren.
Hello, it's such a pleasure to be here and talking about the retina, which is by far the best part of the eye. There's no doubt.
Possibly the body.
Absolutely.
Look, Lauren, we began our journey in vision at the same time, about over 20 years ago now. And then you knew very early on that you wanted to work in research. You did a PhD in paediatric optometry, moved on to traumatic brain injury and ocular motor function, and it seems a natural progression now to be intricately involved in the neurosensory retina and vision restoration. Where does your passion come from?
Yeah, it's a very good question. Look, For me, it's very personal. So I've got two brothers. One unfortunately has passed away, but both of them had vision problems as children. So one of my brothers had amblyopia, or for those of you who aren't eye doctors, that's a lazy eye and quite easily treated. So all he needed to do was wear glasses, put a patch on his eye, and his vision came back to normal. And he now, to give you an idea, he now flies in helicopters telling pilots where to put water on bushfires. So his vision is great now. My other brother, unfortunately, had a brain injury at birth, and he was completely blind from birth. And I remember growing up just thinking it was really unfair that some vision problems we could solve and some we couldn't. And so I think from a really early age, I always had that in the back of my head, why is it that the eye doesn't work sometimes? And why is it that we can fix it sometimes, but other issues are much more complex? So when I got the opportunity to get into optometry, first of all, and then to do a PhD, really delving more into this issue of vision loss, it was really perfect alignment with my interests.
And, through your work throughout the years, you are answering so many questions for us, and we are so thankful that you are doing this sort of work. Now, the idea of the retina, from what I've read about, dates back to periods even before Christ that we know of or that have been recorded, when different theories were being thrown around of how the eye could see. And there was this extramission theory of vision where Plato wrote in the 4th century that light actually emanated from the eye and seized objects with its rays.
I quite like that. It's actually a very lovely thought, isn't it? Like, we reach out to what we want to see and drag it back, but unfortunately it's not true.
Well, Aristotle, the later Greek philosopher, the father of philosophical reasoning, he rejected that. And he advocated for the intromission idea, which is that the light rays were being received by the eye rather than emitted. And so can you believe, though, that these two theories were about absorbing and emitting rays? They were contentious over many centuries. Isaac Newton, another well-known scientist, not credited with discovering the retina per se. His work was across many areas of physics, including optics, but he was fascinated by light and visual and colour perception. He performed a number of experiments on himself, and I'm told that it is quite normal to be performing experiments on yourself as a scientist. The topic for another day, perhaps, Lauren.
Oh, many hours of stories there.
I would probably discourage these things from being performed today. Now, Isaac Newton records that he took a bodkin and put it betwixt his eye, and pressing my eye with the end of it, there appeared several white, dark and coloured circles, which circles were plainest when I continued to rub my eye with the point of the bodkin. Ouch. And if I held my eye and the bodkin still, though I continued to press my eye with it, the circles would grow faint and often disappear until I removed them by moving my eye or the bodkin. That's interesting, isn't it?
So the technical term for what he would have seen is phosphenes. So they're little spots of light. And absolutely do not put a knitting needle in your eye, everyone listening at home. But if you want a more safe way of doing it, you can close your eyes and really gently press on your eyelid, and you'll get the same kind of experience. So that there's little flashes of light. They can be different colours. And what's happening is we're actually changing the pressure in your eyeball, and that's pushing onto the photoreceptor cells in the retina and making them fire. That's why we're seeing light and colour.
So you're stimulating these photoreceptors mechanically. But if he held it there, that effect would disappear. Do you want to talk about what's happening there, Lauren?
Yeah, absolutely. And look, at the risk of jumping forward a few episodes, look, it is a really interesting phenomenon because it's showing that the retina itself is pretty clever in how it works and how the brain interprets what comes from the retina. And so this is a phenomenon called phosphene fading. And so basically what happens is when there is stimuli, which is the same, at some point the eye and the brain go, oh, that's no longer important, I'm going to phase that out. And it's a really interesting thing because when we talk about bionic eyes, which we will talk about in a later episode, these are devices where you use electrodes to stimulate certain parts of the retina. And we find the same thing. If you put electrical current at certain parts of the retina, at some point, the light that the person perceives will fade away because they're still getting that same stimulus. So the retina and the brain go, it's no longer important, I'm going to fade that out.
So I'm hearing now that there's actually multiple ways really to stimulate neuronal retinal cells. So Isaac Newton's done it mechanically. You've just talked now about the retinal cells being stimulated electrically. And then light waves are stimulating our retinal cells also. So there's this cascade of chemical processes that take place once these cells are stimulated. And it moves through multiple layers and cells to be transported to the visual cortex of the brain for visual processing and perception. Let's talk about what are the most important features of this neurosensory retina for electrical signaling and processing?
Yeah, absolutely. So I guess one of the first things to think about is the fact that the retina is actually a very bizarre tissue. So it's actually flipped from the way that you think it should be. So the cells that receive and translate the light into electrical messages are actually right at the back of the retina. So the light that's coming into your eye has to pass through a whole lot of other cells, actually 10 layers in total, until it gets to those photosensitive cells. We can talk a little bit about the different cell types. When you think about, what's obviously they're all important, but one of the most important components are the photoreceptor cells. They're the ones right at the back of the retina. And what they do is when the light hits them, they have a chemical reaction, which then sparks this electrical circuit, which is then passed through the rest of the cells all the way back to the nerves. Photoreceptors are really, really important cells. There's different subtypes. So they react to different types of and different colours of light. And one of the big things with them is that they're very high energy users. As you can imagine, that's a big process.
Yes, because, if you think about vision and seeing and responding to light, it is a continuous process, isn't it? It just does not stop when you have your eyes open.
Yep. Absolutely, absolutely. And the other really interesting thing with these cells is that they don't get replaced during our life. So with the retina, the cells that you're born with are the cells that you have. And that's why eye disease and retinal disease is so devastating, because if you lose these photoreceptors, at the moment, we can't give them back. But what the photoreceptors do to keep healthy and to keep their youth is that they shed what are called the outer segments of the cells. And so by getting rid of those, they're able to grow new outer segments, which then allows them to keep doing that process over and over again. Because you're right, Leanne, there's different light settings. Sometimes you might be watching your child's soccer game. The next thing you're looking at the stars at night. Very dynamic. They've got to do a lot of work.
Completely. So the cells are never replace themselves. It's just that portion of the cells, the outer segments that get replaced. And how they replace? They're replaced by that underlying layer, aren't they? Are they chewed up by that RPE, the retinal pigment epithelium?
Exactly right. So this so it's a very special cell layer, this one. So it is called the RPE because the name's too long for us to say all the time, basically. But it's
not just because we're Australian and abbreviate everything.
That's it, exactly. We'll put like a O at the end or something.
It's global.
The RPE is a single cell layer, so they're very. We can see them when we do imaging, but they're very, very thin layer of cells. And exactly right, they chew up that debris that comes off the photoreceptors, and they're also responsible for bringing oxygen and nutrients through from the blood vessels right at the back of the eyeball through to the retina, because obviously photoreceptors and the other cells need a lot of energy. They need that intake.
Yeah. Let's talk a little bit about the RPE, because that's actually a really important tissue, and it's a tissue that often gets implicated in a number of very popular diseases. So let's talk through the type of waste that starts to accumulate on the RPE. What sort of waste are our cells creating?
Underneath the RPE, so the basement membrane of the RPE is a layer called Brooks membrane. And we know that as someone gets older, that membrane gets thicker anyway, okay? So that happens to all of us over time. But what happens in diseases like age-related macular degeneration, which is the most common retinal disease, you actually get built up at that layer of debris from, for example, the photoreceptors shedding those segments, and also lipids and proteins and all sorts of junk just gets accumulated there. And the problem with that is as all of that junk accumulates and that layer gets thicker, it means you've got this physical barrier in between the blood supply at the back of the eye and the retina. So it's then very hard to get the oxygen and the nutrients from the blood vessels through to the retina where they're needed. And that then leads to those cells dying, we get atrophy, and then people end up losing vision.
So it's that RPE layer that gets fed by that underlying vascular system, that vascular tissue, what we know as the choroid, isn't it?
Mm-hmm.
That starts to be affected by that accumulation of debris because of everything that's happening within those two cellular layers, really, the photoreceptors and the RPE.
And it's just very interesting too, Leanne, because I think quite often when we think about retinal disease, we think about the whole retina and what's happening to the retina in the larger area. But quite often a lot of these diseases start just at that layer. It's that RPE, that single cell layer at the back of the eye. If there's dysfunction there, it then causes damage to the rest of the retina.
Yep. Now it's pigmented. Why is it pigmented?
It has different, obviously pigmented molecules within the layer. One of the really interesting ones is a molecule called lipofuscin. And so we'll talk a little bit later about imaging of the retina, but that's a way that we can use very special imaging techniques to identify where the RPE is and whether or not some of those cells are moving around the retina, because we can look at the different pigments that are present.
And there's speculation around the idea that this pigment is there because it absorbs extraneous wavelengths of light. Is that still the current thinking, that that is one of its functions?
Yeah, absolutely. So I think when you think about it, the light that we get into our eye, there's too much if you think about it, and in particular, interestingly, we get too much blue light into our eyes. And so there needs to be ways to soak that up. And so, the cells in the eye, such as the RPE, are very good at doing that. And the eye's anatomy is very good at doing that. So we have ways that red and green light, for example, is prioritized over blue light in the different cells. And that helps us to have really good daytime vision, because that's what humans really tend to need. We often sleep, unless you're my children, you sleep at night. And so having that daytime vision should be prioritized, and that's what the eye does.
Let's talk a little bit more about this blue, red and green light. So because I think it is really interesting, and over the last decade or so, blue light has become public enemy number one.
Mm-hmm.
Talk us through that, Lauren. Like you've talked about red and green being prioritised by the retina. What do you mean by that?
Yeah, so it's an interesting one. So this is, for those of you, if you haven't heard about this debate, I'm quite impressed. So it is very much around computer screens and, the blue light that we have in our artificial lighting in the world. And there has been some debate about whether that can cause eye diseases like macular degeneration. And so there's been a lot of work going into this. And so one of the things that frightened scientists a number of years ago was that in some animal studies, it looked like if you gave too much blue light, you could cause retinal disease to happen more quickly. Since then, it hasn't been shown in human studies. So we're not able to replicate that in humans. And that's not surprising. Our retinas are very, very different to the retinas of a mouse. And so that's not a surprise. And so there's actually been a recent study done by a group at Melbourne University looking at whether or not we need to think about things like blue blocking lenses and all of these different innovations. And they found that at the moment, the evidence doesn't support that.
Yeah, right. And you mentioned red and green are prioritised. How are they prioritised?
So there's three different types of, there's multiple different types of photoreceptors, but we can think about them in terms of cone photoreceptors and rods. And the names just actually have to do with the shape of them. So the shape, basically a cone photoreceptor looks like a cone. A rod is a longer, straighter, rod-looking structure. Wasn't too inventive with the naming of these ones. But
I like it.
Yeah, exactly. It's nice in science when things are actually straightforward.
Yes, they make sense.
So with the cone photoreceptors, we've actually got three subtypes. So there's small, medium and long wavelength cones. And the cones are just more sensitive to light of certain wavelengths. And so the beautiful thing with that is it means that our retina has a different ratio of those different cone photoreceptors depending on what we need, really. So they're very much aligned with what human vision needs to do. And an interesting space with this now is that we can actually now image the retina at single cell resolution, and we can identify which of the cones are those 3 subtypes. So there's work going on now where you can actually look at the ratios and count the number of the different photoreceptors in the eye.
Many of us are familiar with some basics about the retina, central areas where there are lots of cones concentrated, and then more peripheral areas where there are more rod photoreceptors and many less cones. Let's talk through this distribution and what this means for our vision.
Absolutely. So it really comes back to the function of the photoreceptors. So our cone photoreceptors are really good for our daytime vision, for central vision, high fidelity sight, whereas rod photoreceptors are more useful for nighttime and low light levels. And so roughly speaking, across the retina, you have more cone photoreceptors at the center of the retina and more rods out at the edge. And that's really relevant when we start to talk about diseases, because there's certain diseases that target the rods first. And so then you lose your side vision or your peripheral vision first, versus those that attack the cone photoreceptors first, where you lose your central sight first.
Right. And so you've framed it in a way that there are certain cells which are targeted in certain disease processes. Is that what we're seeing at the moment?
It is, yeah. So, and it's an evolving space. So, historically, back in Newton's time, we wouldn't have known which cell type went first. But we're now getting to the stage with our technologies and our understanding of disease mechanisms that we can work out, which is the cell that goes first and what's the downstream effects of that. Because the retina, when it works well, it works really well. But when one part of that process fails, you usually then get death in the other parts of the retina as well.
Yeah. And so the cones are concentrated at this area called the macula in the retina, which is that area that is primed for central vision, for high acuity vision, for excellent colour vision. And there's a whole bunch of other pigments that you'll find at the macula there as well. What's the importance of those pigments?
Yeah, so those are very interesting pigments. They're called zeaxanthus.
I can't say them.
I was going to say.
I to you.
I was going to say I usually write them down. So how about I cheat? They're called the lutein proteins. But basically, these different molecules within the macula give the macula a very yellow-looking appearance. So when an eye doctor looks into the back of the eye, they can actually see the area of the macula as looking a slightly different colour to the rest of the retina. The reason that they're important is it helps with absorption of light when it comes into the eye, and we can actually measure the level of those macular pigments as well. So there's ways that we can see how many of those pigments are present, and that can then also link into different disease stages too.
So absorption of light as in excess absorption of light or general absorption of light?
A bit of both. So yeah, those molecules are very useful, for our standard living, and also as a bit of a backup if you do get excess light into the eye.
Yeah. Speaking of excess light, Isaac Newton conducted another experiment on himself.
He was not really. He was a very smart man, but not very cautious, was he?
Well, what did he do? He stared at the sun.
Yeah, so he was very interested in this concept of after images, and I think everyone will probably be aware of what we're talking about. So this is when you see something bright, and if you close your eyes, you can sort of still see it hanging around. And there's some really cool visual illusions that you can do. So there's one where you look at a dot and then everything else seems to move around the outside. So this is really ways of seeing those after images. And so Isaac was aware that these were happening. So he decided to sit himself in a dark room, because sitting in a dark room makes your pupils as big as they can possibly be.
Sure does.
Great idea. And then he looked straight at the sun. So again, do not try this at home. A really easy way to damage your retina, and it's a very, very bad idea. But what he did discover from doing this is that when he looked at a really bright object and then looked away, he could get that after image. And it's basically to do with the fact that the photoreceptor cells that we were talking about before get bleached. So if you give them a huge amount of light, then they will be functioning at a certain level. And then when you obviously close your eyelids, they continue to give that effect for a short amount of time.
It's actually incredible that he did not go blind from that particular experiment, because it's akin to using a magnifying glass to concentrate the sun's light rays onto something. And he could have caused a lot of damage. And there's a name specifically for it, solar retinopathy, and he could have burnt a hole in his eye essentially in his neurosensory retina. So.
Absolutely. And look, unfortunately, we do still see it, especially around eclipse time. So the hospitals do get a bit of a blip in people presenting with solar retinopathy at that time. And unfortunately, as we mentioned before, if you damage those cells in your retina, they're not coming back. And so that can lead to permanent vision loss. The other one to be aware of is lasers. So, being very, very careful about lasers that you might buy and play around with at home. Looking directly at a laser can do a similar thing.
Yeah. Now, there's a few other layers you mentioned. And, when I read into this topic, everyone's got a different idea of actually how many layers of the retina there are.
Absolutely.
You've mentioned 10. I've read anywhere from 5 to 19. So how does the retina get stratified?
It's a great question. So I guess I'm a clinical researcher, so I should preface that. So, I'm not looking at these cells in the lab. And so for me, the interesting thing is when I take a photo or a cross-sectional scan of the retina, how many different layers can I see? And that we can get 10. So we can see different layers, and they basically relate to the different stages of that vision process. So to take it back quite simply, what we see is when the light comes into the eye, the photoreceptors are activated. They change that light into an electrical signal, and then it's passed back through the retina, so heading back towards the front of the eye through the bipolar cells, through to the retinal ganglion cells, which are like the nerves in the eye, and they take the signal from that retina back to the brain through the optic nerve. There's other cell types that are really important as well, and they're called horizontal cells and amacrine cells, and also glial cells. And at the risk of boring everyone to sleep, I won't go into too much detail. But the really important thing to know with all of these cells is that they all have very specific functions. So they're about supporting the structure of the retina to keep its, structural integrity, as well as getting that electrical signal from the retina through to the brain so that we can see.
Yeah. And I think it is important to know that our high visual acuity at that centre part of the retina comes from the fact that there's more one-to-one cell interactions there compared with the peripheral retina, where you've got your rods and there might be multiple rods going towards one ganglion cell. Is that?
That's exactly right. Yeah, that's exactly right. So if you think about it, so there are hundreds of millions of photoreceptors in the retina. So it's around 150 million cone photoreceptors and around 7 million rod photoreceptors. But you only have around about 1 million ganglion cells that then exit the eye and go back to the brain. So the retina has to pre-process. It can't send all of the information from those cells through. And so you're exactly right, Leanne. So the cells, sometimes they'll go one-to-one. So one photoreceptor cell through the connections will go to one ganglion cell versus some where they have multiple feed-ins.
Yeah. And what we know actually about these cells has changed quite a lot over the years, hasn't it? The ganglion cells in particular, because again, if we step back and we sort of think about the photoreceptor cells and their function, we know that light stimulates them, hits them, sets off an electrical signal, passes through this chemical process onto other cells. But it's important to think about vision not just as the acceptance of or absorption of light, but also defining and understanding what is actually going on. Is there movement going on? Is there at what intensity is that light stimulating our eyes? And these other cells in our eyes do a lot in terms of processing.
Absolutely. And I think you're exactly right. It's a very moving space. Even now we're still finding different subsets of these cells. So for example, ganglion cells, historically, we thought, well, their job is just the messenger. So the rest of the retina is collecting the light, changing it to electrical signal, doing all the processing, and then the ganglion cell just takes that message through the optic nerve back to the brain. But we're now realizing that some of these ganglion cells are light sensitive themselves. And so they have a really important role to play in our circadian rhythms. So if people are, going to sleep and waking up, that's actually partly to do with the light that's being received into the eyes. And even getting even more into the minute detail, there's different subsets of ganglion cells, which if we're trying to stimulate them with electricity, so for example, again, as we'll talk about another episode, using something like a bionic eye, there is work going into, well, how would you change the electrical waveform that you put into the eye so that you can prioritize which of those cells you stimulate. So quite amazing to think about, decades ago, we thought they were just a one size fits all kind of cell. And now we know there's all of these special techniques that they can do.
What we know about these cells of the retina continues to change and evolve. I mean, going back to the really early theories of our eyes accepting or receiving light in order to see doesn't really do the retina justice, because there's just so many aspects of our visual perception in which the retina plays this integral role, like you've just described, understanding the direction in which things are moving, how bright or dark things are, their colour, even our circadian rhythm, our levels of wakefulness and sleepiness rely on a properly functioning retina.
And at the risk of, blowing everyone's mind in the introduction episode, one of the other really interesting spaces now is that there are technologies that are being developed and certain drugs where we can change the function of those cells. So, if the photoreceptor cells die, which happens in a lot of the diseases, what cells are going to react to the light, right? The photoreceptors are gone, therefore it becomes an issue for vision. And so there are particular drugs which can actually make some of the other cell types light sensitive. So they can take over the function of that, which is just absolutely mind-blowing. And these drugs are in clinical trials. These are being tested around the world.
Yeah. And are these injectables, are they?
They are.
Yeah.
They are.
Yeah. And we'll talk more about injectables, because injectables has become a mainstay of treatment for a number of diseases over the last decade or so.
Yeah.
But we're also moving on from injectables as well, which is also exciting. So then the retina is really a piece of neural tissue, isn't it? Mm-hmm. That is, you mentioned from birth, is what we have, mm-hmm. and that's all we'll have. Mm-hmm. So it's really part of that embryonic tissue from the brain. Is that right?
That's exactly right. And I think this is why, again, very biased, but I think the retina is so amazing. It's a part of the brain, that we can actually view. So, when you go to see your optometrist or ophthalmologist and they look into the back of the eye, they are actually getting a view of tissue which has developed in the same manner as your brain, which is quite amazing. And we can see blood vessels and we can see nerves and we can see the different cell types and really get a good understanding of what's going on in the eye health.
Yeah. Now, we touched on the blood supply a little bit already, but it there is a blood supply. There's actually multiple blood supplies to this little tiny piece of tissue that's made up of 150 million photoreceptor cells, right?
Yeah, exactly.
So what have we got there? We've got this internal carotid artery, mm-hmm. which branches out into what they call the ophthalmic artery.
Yep.
And that branches out into your the blood vessels which supply the back of the retina and the blood vessels that supply the front of the retina, because it's that multi-layered tissue that needs a supply on both sides, essentially, doesn't it?
Absolutely. And it's a very fascinating thing. So one of my favourite fun facts about the retina is it actually has a higher energy demand than the brain. So it's a really energy intensive tissue. So it does need a lot of oxygen and a lot of nutrients. So it does need that dual blood supply. And so the other thing that's very fascinating when you think about that is that the retina is very thin, So this tissue that we're talking about is about the thinness and the texture of a wet piece of tissue paper. So it's very fragile, it's very thin. And so the thought that you actually need blood vessels on the top and on the bottom really gives you an indication of how much work it's doing.
You mentioned earlier about the neural tissue being very interesting or very clever, because it stops seeing what it doesn't need to see. And it explains why we can't see those blood vessels overlying that front surface of the retinal tissue.
Yeah, look, it's a fascinating area, and there's some really fun, again, visual illusions that you can do. So there's one where you can hold your thumb out at arm's length and close one eye, then the other, and you can map where your blind spot is. So this is another aspect. So we can't see the blood vessels on our retina, and we also can't see the area of our retina where all of the retinal ganglion cells converge to turn into the optic nerve to go back to the brain. And you're exactly right. It's because the retina and the brain are very clever at realizing that those spots are not important because they are, currently blocked out at all times. And so they can kind of fill in the gaps. And this happens to patients as well. So when people lose vision, sometimes they won't realize, because even if they lose a patch of vision in one eye, their brain and their eye can kind of compensate for it. So they can quite often not realise that there's a patch missing until it becomes larger.
Fills in the gaps, doesn't it?
It does.
Before 1850, I read that the only way that people would look at the retina was post-mortem.
I'm glad we don't have to just do that anymore.
And there's this golden age of ophthalmology, actually. It's listed here as between 1850 to the 1860s, where they had just invented the ophthalmoscope.
Yeah.
That direct handheld instrument that the practitioners use, that we learnt to use when we were students of optometry back in the early 2000s to visualise the what retinal tissue looks like. There's this one-dimensional flat image, and we had a spotlight. It's really a spotlight, isn't it, where you're trying to look at this tiny piece of tissue, but you're getting very, very small parts of this tissue at any one time through that tiny pupil.
Absolutely.
And we've really progressed so much since then, haven't we?
Absolutely, absolutely. And it is quite amazing when you think about it and how game-changing that is, right? So the ability to view the retina in a living person, absolutely game-changing for this space. But yeah, you're right. Since then, we've now got colour cameras, so we can take retinal photos in that sort of same, two-dimensional plane where we're just looking at the image flat, and then we can do cross-sectional scans. So there's techniques called optical coherence tomography, which actually gives you a cross-sectional view through the retina. So you can look at the individual retinal layers in a living eye, and again, look for areas of disease and see which areas are maybe not functioning as well.
Yeah, it's incredible. And then the latest sort of technique that we've spoken about actually recently is OCT angiography.
Yeah. Yep. So this is, again, another game changer. So historically, if somebody had a bleed in their eye, and that often happened with diseases like macular degeneration, one of the ways to see where the bleed had originated from was to inject a dye into the bloodstream of the person. That's usually through the arm. So they'd be given an injection of this dye. It would travel to the eye, and then the dye over time leaks out of the blood vessels in the retina. So that's a really great technique in terms of looking at where that leak happens and how fast it leaks and all of those things. But the dyes themselves made some people sick. So there were people that were allergic to them. And obviously it's quite intensive and invasive.
Very invasive, yeah.
Absolutely.
Yeah.
So this new technique is using this OCT technology and a special format called OCT angiography, where you can actually look at those different blood vessel layers in more detail. So you can look at the inner ones, so the retinal vessels on top of the retina, and also the ones back in the choroid at the back of the eye. And you can do this in different layers, so you can actually look for any areas of dropout, so areas where the blood vessels might be completely missing, as well as areas of bleeding.
Yeah. So OCT, the ocular coherence tomography, it was invented back in 1991. And when we went through university, I don't recall knowing about it. But it was almost at that time starting to become mainstream in ophthalmology.
Yeah.
Is OCT angiography, is that mainstream yet? Is that sort of at that cutting edge?
Yeah, it's getting there, I would say. So it is still, still very early days with it compared to, clinical available tools, but definitely on its way. And I think when you think about OCT, for example, the OCTs that were around in the 90s, if you look back at those images now, they are just so different. Like all you can really see is the retina. You can just see from the top to the bottom, and that's about it. Whereas now we've got these technologies which give us such fine resolution that you can actually see individual cell layers. And there's a even newer form of OCT coming out now, which is adaptive optics OCT. So adaptive optics just means that you use lenses in the imaging system to make up for the aberrations in the eye. So it corrects all of the light that's coming into your eye just so you get the clearest possible image. And adaptive optics OCT is allowing you to see down to sort of single cell resolution, which is just amazing.
That's incredible, considering the number of cells that are out there. I was trying to think of the population of a country with 150 million cells, and that's
absolutely.
That's a lot of people.
It really is. It really is. And it is interesting, you often, every now and again, I stop and actually just think about the complexity of the retina and the amazing fact that it works. When you think about all of these components that have to go into it, it can, it makes it clear why sometimes it doesn't work, unfortunately.
Yeah, absolutely. So it's really because of these tools that we have that we are now able to visualize exactly what is going on inside the retina and what is going wrong. When they invented that indirect ophthalmoscope back in the 1850s, that was the first time that they had the first descriptions of papilledema, central retinal vein occlusions, optic disc atrophy. So being able to see what is going on makes such a big difference to now our understanding of what is wrong.
Absolutely.
And what can we do about it?
Absolutely. I think a perfect example of that is age-related macular degeneration, which again we'll go into a bit detail later. But one of the characteristic signs of age-related macular degeneration is you get these little white spots on the retina in that macular region. And the beautiful thing with OCT is it's allowed us to see that the accumulations, those spots are accumulations of debris, and it's allowing us to see that they can happen at different layers of the retina as well. So that's allowed us now to sub-classify into what's called drusen versus pseudo-drusen. It's really giving us this very finite and detailed knowledge of the disease mechanisms, which then leads to better treatments as well.
Absolutely, because for a very, very long time, we really couldn't do anything about macular degeneration, could we?
That's right. And you think about, for the clinicians that are listening, we're aware that there are now some really effective injections for certain forms of macular degeneration. But they've really only been around for a relatively short amount of time. And so it is quite amazing and exciting to think about what the insights from imaging that we are now learning will mean for future treatments for different diseases.
Yeah, age-related macular degeneration really is in a class of its own, really, because there's so much that's going on, some of which we understand now, and so much that we still are yet to learn. And that's actually why I've devoted a whole episode to macular degeneration. But other things that can go wrong with the macula, with the retina, I should say, vascular issues. I think vascular issues is a huge one. It's the blood supply to the retina, and we've already talked about how vascularized the retina is and how important it is to continuously be able to provide nutrition and remove waste from the retina. So there's quite a few issues that arise when there is a vascular issue.
Absolutely. And one of the, problems with the retina is the retina does try to heal itself when something happens. So if there's an area in the retina which isn't getting enough oxygen, and that can happen in a number of different diseases, what the retina will sometimes try and do is grow new blood vessels into that area. So that's called
a natural body response, isn't it, to any sort of...
Exactly, try and fix that spot. And so this is neovascularization, so it's new blood vessels growing into that region. But unfortunately, they're not particularly good blood vessels that are grown, so they tend to be leaky, they tend to cause bleeding and other problems that sort of connect in with that. But you're right, there's again, a lot of blood vessels in that area. And so you can have areas where the blood vessels stop working, become blocked, you have, vascular dropout. Obviously, bleeding in the eye is a really significant issue because blood has components in it which are toxic to the retina. So if you get blood in the retina, that can cause damage as well. So it's a very, complex space because the blood, we need it there for the retina, but we need it in the right spots.
And I guess any change to the structure of that tissue, any change to the way the cells are aligned is absolutely going to change what we see with that piece of tissue, isn't it?
That's right, exactly. And so I think, it is very interesting thinking about how imaging and the ability to look at those different layers and to look at what's happening in the retina will help us also to start looking at treatments earlier. So, we can actually see when some of the early signs of damage are happening and hopefully get in at a better spot.
Mm. Now, structural issues with the retina, there could be trauma, there could be holes, there could be tears, membrane formation, tumours. We're not actually going to be elaborating too much on structural issues of the retina when we talk through the retina in this series. Very briefly, Lauren, what sort of changes have been happening in the treatment space of these structural problems in the retina?
So there's a lot of research going into better ways of doing repairs. So, for example, retinal detachment, if there's a hole or a tear in the retina, that has to be fixed. And so there are different techniques to do that. And some of the newer methods that are coming out now are utilising robots, for example. So having robotic components in your surgical equipment to sort of help with that very, very detailed surgery. Look, retinal repair surgery is such a beautiful thing to watch. The surgeons are incredibly talented at really fixing these small holes. Also a lot of research in that space into the complications that can happen. So if you do a repair on a retinal hole or a tear, there are chances of getting an infection, for example. And so there's also a lot of work going into that space.
And speaking of infections, that's something else that can potentially go wrong with the retina too. It can be infected. How does the retina get infected?
Yeah, so it's usually because the eyeball itself has been breached. And so a common one, that is, I guess, responsible for the treatments. So, injections into the eyes, for example, you run the risk of introducing a bacteria into the eye if you do an injection, and also surgery as well. But we do see people that get infections in the retina from other reasons as well. You can also get things like parasites and things into the eye too. But endophthalmitis, which is infection of the eye, can be very devastating for people in their vision.
Hereditary issues that affect the retina. This is one of your babies, isn't it?
It is.
It's really interesting because, again, back to 20 years ago when we were learning about hereditary eye diseases and specifically retinal diseases, if we were to spot any signs or symptoms, it was really counselling. That was pretty much all that we could offer to patients.
Yeah.
And for these particular patients as well, we're talking about working age patients. So it's not like age-related macular degeneration where they've had a really good life and then are slowly feeling the decline of aging. It's actually affecting people's livelihoods and their capacity to live a quality life.
Yeah, absolutely. So this sort of, the inherited retinal diseases, or the IRDs as we call them, they're actually the most common cause of legal blindness in working aged Australians. So they are a significant issue. They are rare in amongst themselves, which is a good thing. But you're right that there's been a huge leap forward in how we diagnose these conditions and how we counsel people about what their particular IRD might mean for them. And excitingly, there's new treatments coming out. And so we will go into some detail on them. Things like gene therapy, stem cells, bionic eyes, different types of drugs as well that can really help to slow down the disease progression when someone has a genetic problem causing damage to their retina.
Yeah, and that will be completely unpacked in another episode. And then glaucoma. Is glaucoma classed as a retinal disease?
It's an excellent question. We always debate this still in research, to be honest. And so in my particular area of research, in these inherited diseases, there's an inherited form of optic neuropathy, which glaucoma is an optic neuropathy. It's damage to the optic nerve called Leber hereditary optic neuropathy. And this is a genetic condition. So we often talk about, should that be tied into the IRD umbrella? So look, glaucoma very much is a disease where the ganglion cells and the optic nerve itself is damaged. But as we mentioned before, the cells within the retina are all very intricately involved and attached. And so, we do very much work with our colleagues who are glaucoma specialists and, really keeping an eye on the correlations and potential relationships between those conditions.
It's completely connected. So now we know about the structure of the retina, how it works, and what might go wrong with the retina. What are your thoughts on the progress that we've made in understanding the retina in your last 20 years of research, Lauren?
It's an incredibly exciting space, to be honest. I was actually moving house and found some of my notes from undergraduate years, so 20 years ago, and it's quite amazing, how far our knowledge has come in the space of 20 years. So I think we're getting such a better understanding of how the retina works, what the cell types do and how we might be able to support them. And also the fact that we can do this really high-level imaging of the retina in a living person is just absolutely amazing. But for me, I think one of the most exciting things is the fact that we're now starting to come up with treatments to help these conditions. And if you think about how things have changed over history, this is very much, we worked out that the retina was there. Isaac Newton kindly poked himself in the eye a few times to work out what it did. We then got better at realizing what it looked like and then learnt how it worked. And now I feel like we're really at the time where we're now trying to correct what goes wrong. And that's why I'm so excited about working in this space.
So we've come a really long way from, that time of understanding the retina through the extramission theory, light being like our bodies having an internal light sensor that, emitted something to capture vision, to now understanding what the retina is capable of.
Yeah.
And even our treatments have evolved quite a bit over time, haven't they?
Absolutely. And it's funny, when you look at some of the historical treatments for a lot of these retinal diseases, they were very destructive treatments. So the idea with a lot of the older school treatments are if there's a part of the retina which is damaged and it's causing new blood vessels to grow into that area, then you kind of want to just get rid of that part of retina.
Destroy the naughty kid.
Destroy the naughty bit. Exactly. And so, some of these techniques still have a place, in treatment of some different eye diseases too. But things like laser and surgeries that were sort of aiming to reduce the need for the retina to try and repair itself, which was causing the damage, it caused a lot of issues in themselves. And so it's quite amazing that we're now at a point in time where we have these treatments which aren't as destructive. So they're treatments that can be given earlier in the disease as well, because you're not running the risks of losing vision from the treatment itself.
Interestingly, those destructive treatments still have a place, don't they? I mean, I know in a lot of my own work in remote regions of Australia where we know we may not be returning to see a patient, or if a patient may not be able to see us really regularly for regular routine treatment that may not be as destructive, those laser treatments are still sight-saving. So I think what we used to do was the best that we could do at that point in time and the best for our situation, but it's so exciting to see these new treatments developing and evolving over time.
Absolutely. And I think it's that concept that there's so much more in the wheelhouse of ophthalmologists now and surgeons when they're looking at different treatments. They can target diseases in different ways and really being able to get in earlier, because with any of these conditions, the earlier you can treat somebody, the more chance you have of saving their vision.
Lauren, we're going to end with you giving us your top 3 things in this area of research that we are likely to see convert from research into the clinical space in the next decade.
Absolutely. So I think a big one in the space of retinal disease will be better phenotyping. So that's a big fancy science word, which just means clinical assessment. So I think that we're going to get to a stage where things like, advanced imaging becomes routine, because we will really need to know exactly what's happening in these retinal tissues.
So really mapping it.
Absolutely. So I think that's going to be something that we get more and more involved in. And linked in with that, for some of these conditions, I think genetic testing will become a big part, and we can, again, unpack that a bit more in a later episode. I think another area that in terms of retinal care and things that will become very important is also looking at the way that vision loss from these retinal diseases affects people in a daily setting. And that's something we haven't really talked about, yet on our podcast. We often talk about the downsides of vision loss, and there are a lot, but there's also a lot of ways that we can help people that do have vision loss. So we've got great adaptive technologies. We've got really good aids to help people with their navigation as well. And so I see more and more of that adaptive technology is going to hit clinical practice and become mainstream.
So interesting. Again, back when we were studying, I remember going through low vision clinics and seeing these CCTV cameras over desks costing patients thousands of dollars to try to enlarge images. What my dad does now, takes a photo, uses his fingers to swipe it and enlarge images. And it's become very mainstream now, hasn't it?
Absolutely.
Which is really exciting for people with low vision.
Absolutely. So, things like, the fact that many of our major smartphone manufacturers, they build this accessibility software in so that it's just default. And that's so important, because it just means that information is accessible for everyone, no matter what their level of vision. And that's something that I'm very passionate about.
Yeah. And your last tip?
My last tip, I think it's got to be treatments. Look, we're absolutely on the verge of a tsunami in terms of different options for treatments. We've got gene therapy, stem cells, new drugs coming up. I work in the clinical trial space, and we are speaking to pharmaceutical companies weekly, about new inventions that they're coming up with that they want to start testing. And so that's incredibly exciting for people that have retinal diseases. There's always a caveat that these things take a long time and they're not all going to work, So all of these exciting things that scientists talk about in podcasts like this, it's exciting. It's really groundbreaking stuff, but not all of the solutions will come through. But even if a proportion do, it's going to be game changing.
Absolutely. Thanks so much for joining us today, Lauren. We really appreciate knowing and understanding more because you're providing this information to all our listeners out there. So thank you so much.
Pleasure.
In this episode, we've discussed how much of our understanding of the retina has actually evolved. Hopefully, you're now familiar with how small, delicate, but extremely active this piece of tissue is. That it's not just receiving light inputs, but actually processing these inputs as well in really different ways for visual perception rather than just vision, and even wakefulness. Our knowledge of the retina has really been advanced with technology and different visualization techniques, and it certainly is keeping pace with the dynamic rate at which technology is evolving today. It's exciting, and we'll be discussing more in the following episodes of this series of Talking Retina. Thanks for listening to Talking Eyes. Additional resources for this episode can be found in the show notes. If you found the content of our podcast interesting, feel free to post a great review and share it with your community. If you have a topic you'd like to hear more about, or have expertise in the field of vision and would like to contribute to future series, contact me via our socials on Facebook and Instagram. I'm Leanne Trin, your host of Talking Eyes.